Have you ever tried organizing a completely chaotic computer desktop? The kind with millions of unsorted, randomly named files gathered everywhere? It's an absolute nightmare, right? You'd probably start by making a couple of broad folders, maybe just work and personal. But as you accumulate more and more files, you quickly realize you need subfolders, tags, and a way more sophisticated system just to make sense of it all. Well, biologists have faced the exact same problem with the millions of life forms on Earth. Welcome to today's Explainer, where we're going to dive into Earth's ultimate operating system, the evolution of biological classification.
Here's our quick roadmap for today's Explainer. First, upgrading Earth's filing system. Second, the five-kingdom breakthrough. Third, Monera, the invisible majority. Fourth, Protista, the microscopic misfits. And finally, Fungi, the great decomposers.
All right, let's jump right into Section 1, Upgrading Earth's Filing System, from version 1.0 to 5.0.
Think about how much our technology has scaled up over time. Our scientific understanding has done the exact same thing. Way back in antiquity, Aristotle released what we can call version 1.0. He used very simple, observable physical traits. He divided plants into trees, shrubs, and herbs, and he separated animals into two incredibly basic folders, those that had red blood and those that didn't. By the 18th century, Linnaeus rolled out version 2.0 with the two-kingdom system, Plantae and Animalia. Now, it was easy to understand, sure, but it had some major glitches. It literally couldn't distinguish between simple and complex cells or single-celled versus multicellular organisms. You had green photosynthetic algae lumped right alongside completely different non-photosynthetic fungi. So, in 1969, R.H. Whitaker introduced a massive, much-needed update, version 5.0, the five-kingdom system.
Which brings us to Section 2, the five-kingdom breakthrough, Whitaker's master blueprint.
Whitaker didn't just look at the outside of an organism. He dug into the microscopic hardware to create his rules. He set up very specific criteria. First, by checking for a nuclear membrane, he gave the simple prokaryotes their own dedicated kingdom called Monera. Then, by analyzing the chemical makeup of cell walls, he separated out the plants, which have cellulose, from the fungi, which have chitin. And by strictly looking at the mode of nutrition, he finally clearly separated the autotrophic plants that make their own food from the heterotrophic animals that ingest food and the saprophytic fungi that absorb dead matter. It was a brilliantly logical system.
Actually, the real game changer here, the crucial point you have to understand, is the fundamental difference between simple prokaryotes and complex eukaryotes. This single distinction totally broke the old two-kingdom rules. Prokaryotes, which lack a nuclear membrane around their genetic material, think of it like having your data scattered without a central folder, were all grouped into Monera. Everything else, the eukaryotes, with their well-defined membrane-bound nuclei, populated the other four kingdoms.
So let's open up that first new folder in Section 3, Monera, the invisible majority. Bacteria are the sole members of this kingdom, and they are literally everywhere. I mean, hundreds of them live in just a handful of soil. They're primarily grouped by four basic architectural shapes. You have the spherical cocci, the rod-shaped bacilli, the comma-shaped vibrio, and the spiral spirilla. But listen, don't let their simple single-celled looks fool you. As a group, bacteria showed the most extensive metabolic diversity of any living organisms on Earth.
Let's really unpack that metabolic diversity. Because within Monera, we basically have two very distinct factions. First, we have the extremists, the Archaeobacteria. These guys differ from other bacteria because they have a completely different cell wall structure that lets them survive in the most hostile environments imaginable. We are talking halophiles thriving in extreme, super salty environments, thermoacidophiles surviving in literally boiling hot springs, and methanogens living deep in the guts of cows, which, by the way, are responsible for producing methane biogas. On the other side, we have the builders, the eubacteria, or true bacteria. These are characterized by a rigid cell wall. This group includes cyanobacteria, or blue-green algae, which actually have chlorophyll similar to green plants. Some of these can even fix atmospheric nitrogen in specialized cells called heterocysts. They play a massive role in recycling the Earth's vital nutrients like nitrogen, phosphorus, iron, and sulfur.
As a quick, kind of crazy footnote in the Monera folder, we have the mycoplasma. These are the ultimate rule breakers. They're the smallest living cells known to science. They completely ditch the rigid cell wall that defines other true bacteria, and get this, they can even survive entirely without oxygen. Unsurprisingly, a lot of these tiny outliers are highly pathogenic to plants and animals.
Let's jump into Section 4, Protista, the microscopic misfits.
The boundaries of the Protista kingdom are not strictly defined at all, which makes it essentially biology's taxonomic junk drawer. It's full of an amazing variety of microscopic, single-celled eukaryotic misfits. Take dinoflagellates, for example. These marine organisms possess two flagella, and they can multiply so rapidly they trigger toxic red tides that wipe out massive numbers of fish. Then compare that to the euglena. These are bizarre shapeshifters. They don't even have a cell wall. Instead, they have a protein-rich flexible layer called a pellicle. Even crazier? They act like plants and photosynthesize in the sunlight, but if you plunge them into darkness, they totally switch modes and become tiny predators just to survive.
The gap between organisms in this kingdom is just incredibly wide. In one corner, you have saprophytic slime molds that creep over decaying twigs and leaves, engulfing organic material. Under the right conditions, they form a massive creeping aggregation called a plasmodium that can spread out over several feet. And then, in completely different aquatic environments, you find paramecium. These are highly coordinated organisms covered in thousands of tiny hairs called cilia. The synchronized beating of these cilia actually steers water laden with food directly into a bodily cavity called a gullet.
So, to quickly map out this junk drawer, Protista organizes into 5 distinct, incredibly different subfolders. Chrysophytes, whose indestructible silica-embedded cell walls accumulate over billions of years to create diatomaceous earth, which we actually use in our water filters. Dinoflagellates, those toxic red tide creators. Euglenoids, the flexible dual-mode survivors. Slime molds, the great microscopic engulfers. And protozoans, primitive animal relatives that act as predators or notorious parasites, like the malaria-causing plasmodium.
Finally, we reach Section 5, Fungi, the Great Decomposers. To truly grasp what fungi are, you have to look way past the mushroom cap you see popping up above ground. With the exception of single-celled yeasts, fungi are mostly filamentous. Their true body is actually a vast, mostly underground network made of long, slender, thread-like structures called hyphae. This entire interwoven network of hyphae is what biologists call a mycelium. It's almost like a giant, underground biological internet.
For centuries, humans filed fungi under plants. But that was a huge mistake. Fungi have a completely different chemical signature. They absolutely do not have cellulose cell walls. Instead, their cell walls are composed of chitin and polysaccharides. And because they lack chlorophyll entirely, they are strictly heterotrophic. They absorb soluble organic matter from dead substrates, acting as the world's great saprophytic decomposers, or they live as parasites or symbionts. This unique chemistry completely separates their evolutionary path from plants.
Now, fungal reproduction can seem highly complicated, especially since they can reproduce vegetatively, asexually, and sexually. But their sexual reproduction cycle is basically a brilliant 3-step dance. Step 1 is plasmagamy. This is the fusion of the cellular fluid or protoplasm between two gametes. Step 2 is karyogamy, the actual fusion of the two distinct nuclei. Interestingly, in some fungi, this nuclear fusion is delayed, creating a really unique dikaryotic phase where a single cell just hangs out with two separate nuclei for a while. Finally, step 3 is meiosis, where the resulting zygote undergoes division to produce new, genetically distinct haploid spores inside fruiting bodies.
Based on the structure of their mycelium and exactly how they form those spores, fungi are neatly filed into 4 distinct biological classes. First are the Phycomycetes, found in damp environments. This is where your common bread mold Rhizopus lives. Next are the Ascomycetes, or sac fungi, which is a wild group including everything from single-celled yeast used for baking to the life-saving antibiotic source penicillium to highly prized edible truffles. Third are the Basidiomycetes, which includes the common agaricus mushrooms you buy at the grocery store, as well as destructive parasitic crop rusts. And lastly, we have the Deuteromycetes, often playfully called the imperfect fungi. They get this name simply because scientists have only ever discovered their asexual or vegetative phases. The moment a sexual stage is found, they get immediately transferred to one of the other 3 classes. So, we've explored Whitaker's massive version 5.0 classification system, from the simple, survivalist bacteria of Monera all the way to the complex, networked decomposers in fungi. But science is never truly finished. It's constantly updating. With recent breakthroughs in molecular biology, scientists have actually proposed dividing the Monera kingdom entirely, creating a 3-domain, 6-kingdom system. As we uncover more hidden code in the DNA of life, one has to wonder, how will Earth's biological operating system evolve next, and what invisible kingdoms are still waiting out there to be discovered? Thank you so much for joining me today, and keep exploring.